In this video, we learn about how all of this DNA and RNA stuff translates into the development of an organism. Do you remember what happens to mRNA after it is transcribed but still in the nucleus? Well, you should remember that mRNA is edited. The introns are cut out and the exons are spliced together. So, how do you define gene expression? Well, gene expression is the way that DNA, RNA, and proteins put genetic information into action in living cells. So, based on what you know, how do you think mRNA rather, how do you think that mRNA editing affects gene expression? The answer to that question is that sections of mRNA are cut out and they are not translated into proteins. Therefore, only the parts of the gene that are left are expressed. So in this lesson, we're going to learn about other ways that gene expression is controlled. All right, so we're going to begin with prokaryotic genes and how they are regulated. Now, the DNA binding proteins in prokaryotes, they regulate genes by controlling transcription. So in order to conserve energy and resources, prokaryotes regulate their activities using only the genes that are necessary in order for the cell to function. And by regulating gene expression, bacteria can respond to the changes in their environment. For example, the presence or absence of nutrients. So how does an organism know when to turn a gene on or off? Well, one of the keys to gene transcription in bacteria is the organization of genes into operons. An operon is a group of genes that are regulated together. The genes in an operon usually have related functions. The E. coli that we see here in this image, as an example, has operons within its DNA. So E. coli are bacteria. The 4,288 genes that code for proteins in E. coli include a cluster of 3 genes that are regulated together for the metabolism of lactose. Now, lactose is a sugar. We get it from dairy products like milk. And if you don't have the enzyme to metabolize lactose, you have what's called lactose intolerance. To digest lactose, the E. coli needs these certain enzymes. The 3 genes that code for such enzymes in E. coli are called the Lac Operon. So why does E. coli have to be able to switch the lac genes on and off? Well, lactose is a compound that's made of 2 simple sugars, galactose and glucose. To use lactose for food, the bacterium has to transport the lactose across its cell membrane and then break the bond between glucose and galactose. So these tasks are performed by the proteins that are coded for by the genes of the Lac Operon. So this means that if the bacterium grows in a medium where lactose is the only food source, it has to be able to transcribe these genes and produce these proteins. If grown on another food source, such as glucose, it would have no need for these proteins. Now, the bacterium is able to detect the presence of lactose, and therefore, when the products of these genes are needed, they can be used. When lactose is not present, the lac genes are turned off by regulatory proteins that bind to DNA and block transcription of the lac operons. Now, on one side of the operon's 3 genes are 2 regulatory regions. There's a promoter region and an operator region. The promoter is a site where RNA polymerase can bind to begin transcription. It's right there. Now, the operator site is where a DNA binding protein known as a Lac repressor can also bind to DNA. And we see the oppressor, or repressor, I should say, right there. Or repressor. We see the repressor right there. When the repressor is bound to the operator, RNA polymerase can't move along the DNA and reach the genes. In other words, the repressor is like a roadblock. The gene that codes for the repressor protein is located before the promoter region, right there. So what is the function of the repressor? It can bind to the operator, blocking RNA polymerase and stopping transcription and therefore gene expression. Notice that the Lac genes are over here, further down the RNA. Notice that the Lac genes are down here, further down the DNA. Okay, so looking at the same segment of DNA in an E. coli bacterium as what we saw on the previous illustration, just keep that in mind because all of the parts are the same. We have the repressor, we have the promoter region, we have the Lac Operon out here, we have the genes here. All right, so, and we have the RNA polymerase. So that's what we have being shown here. We got the repressor that's actually bound to the operator, and it's blocking RNA polymerase from transcribing those genes. So if this is the case, is lactose present or absent? Well, in this case, it would be absent, which is why the repressor is blocking transcription. The repressor binds to the operator, preventing the RNA polymerase from binding to the promoter. See how it's kind of in the way? Yeah, so RNA polymerase can't do its job. All right, here again, looking at the same segment of DNA in an E. coli bacterium as the previous two illustrations. So if the repressor protein is always present, how can the Lac genes ever be switched on? Well, the Lac repressor protein has a binding site for lactose. Okay? And here we have lactose present in this particular illustration. So what happens when lactose is present? Well, the lactose binds to the repressor, and this causes a change in the repressor structure. With the lactose bound to it, the repressor can't bind to the operator, and this will allow the RNA polymerase to bind to the promoter. When lactose is added to the medium, it diffuses into the cell and attaches to the Lac repressor. This changes the shape of the repressor protein in a way that causes it to fall off the operator. Now, with the repressor no longer bound to the O site, RNA polymerase can bind to the promoter and transcribe the genes of the operon. So how is the way that lactose turns genes on and off to the way that cold air signals a furnace to turn on or off? Well, cold air causes a furnace to turn on. When the air is no longer cold, the warm temperature causes the furnace to turn off. Lactose works in a similar way. The presence of lactose causes Lac genes to turn on. When the lactose is no longer present, the absence of lactose causes the Lac genes to turn off. Now let's take a look at eukaryotic gene regulation. The general principles of gene regulation in prokaryotes also apply to the eukaryotes, but there are some differences that we're going to point out. Eukaryotic genes are controlled individually, and they have a more complex regulatory sequence than those of the Lac repressor system, for example. The illustration you see here shows what's called the Tata box. One of the most interesting features of a typical eukaryotic gene is that Tata box. So the Tata box is basically this short region of DNA. It's about 25 or 30 base pairs, and it appears before the start of a gene. and it contains the sequence TA, or TATA. The TATA box binds a protein that helps position the RNA polymerase by marking a point just before the beginning of a gene. So, getting transcription started, which is shown in this illustration, is just one way that eukaryotic genes can be regulated. They can also be regulated by transcription repression. So, how are genes regulated in eukaryotic cells? Well, by binding the DNA sequences in the regulatory regions of eukaryotic genes, transcription factors are able to control the expression of those genes. So, gene expression in eukaryotic cells can be regulated at a number of different levels. One of the most critical is at the level of transcription by means of DNA binding proteins known as transcription factors. Some transcription factors enhance transcription by opening up tightly packed chromatin, while others help attract RNA polymerase. Still, others will block access to certain genes, much like prokaryotic repressor proteins. So, why is gene regulation in eukaryotes more complex than in prokaryotes? Well, it's because of the way that genes are expressed in a multicellular organism. Complex gene regulation in eukaryotes is what makes differentiation and cell specialization possible. So consider the following. Genes that code for, say, liver enzymes aren't expressed in nerve cells, and keratin, which is an important protein in skin cells, that's not produced in blood cells. Cell differentiation requires genetic specialization, yet most of the cells in a multicellular organism carry the same DNA in all of their nucleuses. So, what controls the development of cells and tissues in multicellular organisms? As an embryo develops, those different sets of genes are going to be regulated by transcription factors and repressors that bind to DNA. So these, in turn, will regulate transcription and the production of RNA molecules. So regulation of both DNA and RNA in this way helps cells undergo differentiation, becoming specialized in structure and function. Now, American biologist Edward Lewis did some really good, really solid work, and he was able to make it clear that a set of master control genes known as homeotic genes regulate body parts that develop in specific parts of the body. The molecular studies of homeotic genes show us that they all share very similar 180 base DNA sequences. And this was given the name Homeobox. So, Homeobox genes code for the transcription factors that activate other genes that are important in cell development and differentiation. In flies, for example, there's a group of Homeobox genes known as Hox genes, and they're located side by side in a single cluster. Hox genes determine the identities of each segment of a fly's body. So, what section of the bodies of flies and mice, for that matter, are coded by the genes shown in blue here in this illustration? Well, the back portion of the body, the posterior portion of the abdomen of the fruit fly, and the rump of the mouse. Organisms can also express certain genes only during particular environmental conditions. Caterpillars, the pupa, and the butterfly, they all have exactly the same genes. The coloring of the coat of the Himalayan rabbit is another example of the effect of the environment on gene expression. So, what would happen if a cold pack were strapped to a Himalayan rabbit's back while its fur was growing in? Well, if that cold pack was strapped to a Himalayan rabbit's back while its fur was growing in, the fur under the cold pack would grow in black.